The talk today is mainly due to two people, I guess. John, for constantly telling me that targeted therapies are not the way forward and early detection is. And Harpal Kumar, who once I built a cancer biomarker center a few years back, said, well, you're doing the easy bit, Caroline, but why aren't you doing early detection? And given he was in charge of Cancer Research UK, who'd been funding me since I was about 12, I felt that perhaps we should move in that direction for all the right reasons. So it's easy to say, it's actually really difficult to do. So what I want to do today is tell you about the progress we've made, review the field a little bit, and talk about liquid biopsies for the earlier detection of lung cancer, the challenges and the way ahead. And the alternative title of the talk is, are we fishing in the right pond with the right fishing rod? And the fish over here, I hope you can see my pointer, are all the things you could measure in the blood. And the question is well, apart from these, which are measured in the breath, but you know, the really big question still to be tested and shown robustly is what is this liquid biopsy that picks up cancer early going to look like? So those are my disclosures. And I guess I was going to start with some of the very many challenges. I think it goes without saying that we all think early detection is the best way to change outcomes for patients. It's what's going to really transform cancer patient outlooks. But it is tricky. So question first of all, is it even possible with the technologies we already have? The assays need to be sensitive and specific. We're worried about false positives, telling someone they have a cancer when they don't, false negatives, missing those early cancers. And what we really need is assay validation that is extremely robust. And the problem with that statement is you have to do an enormous experiment to get to that point. Assay testing, where do you test the assay? And you start with the detection of early cancers retrospectively. But that's a very different challenge from the early detection of cancer prospectively. So people are beginning to think about minimal residual disease, for example, after surgery of an early cancer, as sort of the low-hanging fruit of a test bed for an early detection assay, because there are knowns instead of unknowns, in much as you've taken a tumor out, you can sequence it, you know what mutations you might be looking for, as opposed to early detection of cancer where you really don't. But it will give you an idea of whether you have a sensitive test. So these are sort of the approaches people use when they're beginning to develop a liquid biopsy for the early detection of cancer. I'm going to talk pretty much exclusively, once I get going, on the data on lung cancer. And the question for the perhaps wider cancer population is, do you look at everybody? Is this a whole population test? Or do you go first for the high risk? And that's really like fishing in an ocean versus fishing in a pond. And in lung, of course, we know who the high risk are. These are the smokers. But those are the very folks who are perhaps going to be less, they won't come forward quite as much. They have low participation in screening studies. And we know this, and it is really all tied up with societal and lifetime factors. And of course, there are an increasing proportion of folks who get lung cancer who are non-smokers. So those are the things we have to think about. The other thing we have to think about, particularly in some other tumor types like prostate and breast cancer, for example, is overtreatment with potential for harm. So we can detect early, let's say, but do we know whether we have an indolent disease or a potentially lethal disease? And we do have challenges with the resolution of imaging, which I'll come back to. Are we looking at clinically observed disease versus biologically defined disease? And we always have to worry about the worried well. So if you do early detection in the UK in Cambridge, you're going to come across a lot of the worried well. If you do early detection in the northwest of the UK, in Manchester, for example, you're going to be looking at these folks who are unlikely to participate in screening. So very, very different demographics. Sample acquisition is critical. Where do you do this? Do you do that in primary care? I think it's better to base this in a community, and I'll come back to that. You've got to look at the feasibility if you're going to do a community-based analysis, but it really should be non-disruptive because you want high compliance. And I'll come back to all of those issues. You know, and if you do it well, you might get more lead time, and this may apply in lung cancer, but if there's no curative treatment, how much further forward are you? So if you can do it well and have surgery with a good chance of cure in lung, that clearly is the way to go. But what about targeted therapies for earlier disease? You know, and this is where minimal residual disease monitoring trials are coming in. with earlier interventions using targeted or immunotherapies. And again, we can discuss how likely they are to be helpful down the line, but actually I don't think we really know. What we do need for this is team science. So you need large multidisciplinary teams. I think you probably need international efforts. The study size and length are a challenge for a 3-year postdoc, for example, because they're long-term projects. So, you have to think about who is actually going to do them. And of course, one of the things that's just prohibitive is cost. So who pays? And I think one of the ways forward is a private-public partnership. I'll talk about Grail briefly and their relationship with the National Health Service in the UK and the big trials they're setting up. And I think what is beginning to happen is the health economics of this. If we got a liquid biopsy that really works for early detection of cancer, how much money would that save national health, like someone like the NHS in the UK and more globally? So those are some of the very many challenges. And just to sort of carry on with that, which subjects should we prioritize? The high risk, looking in areas of social deprivation, the poorly educated who make those bad lifestyle choices. Where and how do we collect the sample? Is there a sweet spot for intervention? So will we detect at a threshold below imaging resolution? What about those small tumours that we might find that need a zip code? And that's where methylated circulating tumour DNA comes in. Risk of relapse tests. So how do we, if we found it early and we've done surgery, what do we do next to see if we can catch those folks who may relapse despite the early detection? And how do we profile for that early personalised intervention? And this is one of CRUK's graphics for tumour progression. And I think we have to challenge it a little bit. Is it linear like this? Or do we actually find that even in early disease, you'll see cells that have shed or invaded, entered the blood vessels? And do you see these circulating tumour cells actually quite early on? And I think there's lots of evidence now to suggest that you do, and I'll show you some for lung. Again, I'm keen on a community-based effect, a good compliance protocol for early detection with minimal disruption to your regular day, particularly if we move to a whole population type screen. We always have to think about pre-analytics. So once you take that blood sample, it's got to be portable, it's got to be stable. And are you going to go to a centralized testing site? And in which case, if you have several of those, you're going to need cross-site validation, for example. So again, the who, where, and when of early detection. And this is really who we need in the team. So you can go through this: bioengineers, nanotechnologists, molecular and cell biologists and analysts, various types of clinician, epidemiologists, radiologists, statisticians, health economists, psychologists, bioinformaticians, data and computer scientists, quality of life specialists, patient and public advocates, absolutely essential, community health carers. I'm quite keen on the van drivers and the policy makers. And I probably haven't even named them all, but this is the kind of multidisciplinary team you have to pull together, I think, to do early detection well. And I guess the other thing that's happening, and it's taking a bit of time, but I think there's momentum now in a way that there wasn't previously, is the private sector culture change and the incentive for early detection. I think it is beginning to happen, and I think Grail is just one example of that happening. But getting big pharma on board for early detection, I think, still is quite a big challenge. So let's get to what we need to measure. So you might argue, and I'm not an immunologist, but I think there's reasons to argue that the innate immune system would be the earliest responder to a tumor in a human body. And the question really then is, what is the specificity? And how could you measure it anywhere other than in the tumor microenvironment, which is a non-starter really for early detection of most tumor types? But something we're beginning to think about, and many people across the globe, is there something in the blood? Are there types of immune cell in the blood, that in the periphery, that can give an indication of a presence of an early tumor? Most of the action is in circulating tumor DNA. And the big question in lung, at least, is can it really get sensitive and specific for stage 1 disease? And the other question, I guess, is does it need to? Is it going to be sensitive and specific enough? We always have to worry about clonal hematopoiesis of indeterminate potential. I'll come back to that. We're going to need these methylation postcodes, I think, and enormous experiments are underway, and Grail just being one of them. There's some really nice work coming out of Italy on circulating microRNA. You only need a tiny sample of blood. I think it's really promising, and those big prospective validation trials in large cohorts are underway now. I'm a bit of an addict to circulating tumor cells, and I will talk about those. So they give you genotype and phenotype. And the question, of course, was, and I think remains, is it too late once they get out and about? So I'll come back to that. We can look at proteins. Plasma mass spec has been sort of bedeviled. It's promised much and delivered little so far. I think the technology is becoming more adept now for early detection. We just need to figure out what we do, those high abundance proteins in the blood. There are some tests based on autoantibodies. I've not been overly impressed with them so far. The new kid on the block is tumor-educated platelets. Apparently they can give you RNA signatures out of a drop of blood as well as postcodes. I think more work needs to be done and published, and cross-site validation critically important there. We should perhaps not forget the sniffer dogs, and clearly there are in lung cancer certainly early detection breath tests being developed. We have imaging, of course, and I'll talk about it, but the resolution is really not good enough. There are lots of false positives for low-dose CT scanning, and there are lots of indeterminate nodules in the lung which cause big problems. And in the end, do we need a mixture of all of the above in a multimodal test algorithm with machine learning and all of these AI approaches, which are really quite difficult for most of us to understand? But I think what is also clear is we do still need more knowledge of early biology to guide analysis. So we can do our fishing trips, but wouldn't it be great if we had fantastic preclinical models, patient-faithful preclinical models of early disease, so that we could actually have an informed idea of what we should be looking for in an early detection test. Can't really talk about this without the crux of the matter, which is sensitivity and specificity. So this graphic, which I won't dwell on, is really what it's all about. Need a cut point with high specificity and sensitivity. But I think when you talk to most people who are really getting really deep into early detection assay development, specificity really trumps sensitivity all the time. So you need your sensitivity to be as low as possible, but if you lose too much specificity, your assay is not going to be helpful. And of course, the earlier stage disease, the lower your signal. So if we think about ctDNA, this is a historical slide. It's been known for a long time that not all tumors are good shedders of ctDNA. It's also been known for a long time there are very few copies of ctDNA in patients with early stage cancers. And these are some of the early papers that I won't spend time on. But this is where it's really been important to get ctDNA methylation and better mutation sensitivities to take this forward in lung. And I just wanted to mention the lung clip assay from Max Dean in Stanford, who's a collaborator of ours, and we have a multimodal assay development ongoing. And he really developed his assay from a tumor-informed analysis, which first involved sequencing tumors, early stage tumors, to identify mutations that might be helpful to track in plasma that could be used as an early detection test. So once he'd done that, it was very clear that in early stage non-small cell lung cancer, the levels are very low. The median was 0.03% ctDNA in plasma for stage 1, 0.1 for stage 2, and 0.5 for stage 3. So this is the level of sensitivity that is really, really challenging. So what he started to do next was build a tumor-naive ctDNA based lung cancer early detection assay. He looked at 104 early stage non-small cell lung cancers with risk matched controls and started to train a machine learning based model that integrates tumor mutation and copy number to identify in a plasma sample which plasma sample is likely to contain lung cancer ctDNA. So it's a beautiful study. It's published in Nature last year. And I guess for me, this is the most sensitive mutation assay for lung cancer on the block at the moment. It's got promising performance. Sensitivity, you see 41 to 67% for stage 1 to 3. So really not still sensitive enough, but a wonderful specificity. So we're working with Max, and I'll come back to it, and Peter Kuhn at University of Southern California with my team together to look at a combination to see if we measure methylated ctDNA and circulating tumor cells and lung clip together, will that give us more sensitivity and more power? And just to pull out some of the things that Max showed with the lung clip, You can show that the CTDNA he picks out are correlated with stage, as you might imagine. The CTDNA levels are prognostic within stage 1, and what that suggests is if you have a stage 1 tumour that is likely to progress and be very aggressive, there's where you're going to see the higher levels of CTDNA. This is just the schematic of his integration and machine modelling for copy number and mutations, and the model performance by stage is in an independent validation cohort of 46 cases and controlled compared to the training cohort in blue. So this is perhaps the most promising that's published, at least from an academic group. It's worth just spending a minute on the fish and chips. I always think about this as fishing, so this is the analogy. Works well in the UK, not necessarily everywhere else, but deep targeted sequencing of white blood cell controls are really important if we want to really think about this clonal hematopoiesis of indeterminate origin, or CHIPs. And this is data that Charlie Swanton presented a few years back at ASCO. So it's the predictive chip burden, 6.5 variants per individual at the age of 65. So this is a really big deal, and you have to control for it when you're doing your early detection studies. This is straight from the Grail website. So they're developing a blood test combining high intensity genomic sequencing, complex computational algorithms, bit of smoke and mirrors there, vast amounts of clinical data to find cancer specific patterns. They've called their test, which they're going to now trial in the NHS in the UK, Galleri, and the government here and Grail are going to work together for this. This is a multi-cancer, so it's not what we're doing, which is fishing in the pond with the high-risk lung cancer folks. This is multi-cancer, everybody, early detection blood test. And that's about to start this summer, kicking off. If we look at one of their earlier papers, and again, just pick out the highlights from this paper in Annals of Oncology, which is a sensitive and specific multi-cancer detection and localization using methylation signatures in cell-free DNA. Thousands of participants, 2,482 cancers, more than 50 cancer types, training and validation sets. Their specificity is really good, 0.7% false positives, but the stage 1 to 3 sensitivity, 67.3, in 12 pre-specified of the 50 cancers, and only 39% in stage 1 disease. So either they've done something better that we don't know about because it hasn't been published in Galleri, or they're looking at, perhaps not really great sensitivity in stage 1. So it's going to be really interesting to see how the NHS trial with Galleri pans out as we go forward. Really exciting stuff, and I guess the proof will be in the pudding. I'm just going to show you one slide from tumor-educated platelets. This is the Verninger paper from BEST in 2018. I think they've published again on TEPS, but not many other folks have published on TEPS that I know of. I mean, there is obviously a company behind all of this, but they're talking about a drop of blood. They can look at growth factors, cytokines, exosomes, nucleic acids, extracellular matrix, and reception interactions. There's all sorts of things they can do in one drop of blood. And I think for me, very exciting stuff. I'd like to see other groups produce the kinds of data that Verninger has. Not that I doubt him, I think he's a fantastic scientist. I just would like to see more groups do it. The bioinformatics is incredibly complicated. So let me go to what we're trying to do in Manchester now to detect lung cancer early. So we know that up to 50% of non-small cell patients who have surgery with curative intent relapse. And you could hypothesize then as if circulating tumor cells are the architects of micrometastatic disease, which we believe they are, and we have some data for that. Can CTCs predict risk of recurrence at the point of surgery? And if this is so, could it inform on the frequency of minimal residual disease monitoring? And more pertinent for today, if they're out and about early, can we use them for early detection? So I guess it's worth just looking at this to understand the challenge of rare cells in the blood. Generally speaking, for most tumor types, 1 CTC and 1 ml of blood. So it really is the needle in the haystack problem. And this is what else is in the blood, because, guess what? There's blood cells there, and there's a lot of them. There are white ones, there are red ones, there are platelets, and you can see that the CTCs are extremely rare. So this was the pilot study that got me excited in terms of CTCs as early detection biomarkers, and I'll take you through a series of data slides that will come back to the early detection question shortly. But this is not looking in the peripheral blood sample, this is looking in the pulmonary vein when patients with early stage non-small cell lung cancer 1 to 3A come for surgery. So what happens is the tumor is about to come out, and just before it's removed, you can take a blood sample from the draining vein of the cancerous lung. And this was with cell search. So it's worth pointing out that cell search is a terrible technology for non-small cell lung cancer, actually, because it only finds epithelial cells. And I think what we're learning as we go is actually it's the mesenchymal cells, probably the cells that are undergoing EMT in non-small cell lung cancer, that are more prevalent. But nevertheless, with this suboptimal technology platform, you can see, you see very, very few CTCs in the periphery, almost never, certainly almost never in stage 1 and 2 disease. But there are patients with stage 1 disease, and there are certainly patients with stage 2 disease for where you can find epithelial cells. Now, we didn't sequence them, so we can't be sure that they're CTCs, but they're epithelial cells, and it's rather odd that they're there. And in this very small pilot study that got us going, if you see those CTCs in the pulmonary vein, this is disease-free survival, you have a higher chance, perhaps not surprisingly, of having a relapse of your disease. So what this began to suggest was that cell search CTCs are out and about early, that these pulmonary vein CTCs may predict the risk of recurrence. This was only a 32 patient pilot study, and this is Phil Crosby, the thoracic physician that we work very closely with on our early detection studies. And what we went on to do next was validate it in the TRACERx National Lung Cancer Consortium led by Charlie Swanton. So just to give you a quick introduction to TRACERx, this is a study of 800 plus patients now. We're coming to the end of TRACERx, in fact, where patients come for their surgery and then we follow them up all the way through. Charlie looks at the resected tumor with multi-region sequencing. We get a metastatic tumor, and this is going quite well, not from everybody, but from a significant number. And my job in TRACERx is to look at CTCs and CTDNA. So this is the pulmonary vein blood collection at resection, and then obviously peripheral blood collections longitudinally thereafter. The aims of the program as a whole, which is running across the UK, is to define lung cancer evolution through multi-region and longitudinal tumour sampling and genetic analysis, to determine the impact of clonal heterogeneity on clinical outcomes. From this point, from the point of view of this talk, to predict the risk of recurrence and to monitor mineral residual disease and ultimately novel therapeutic targets, which is something many in this audience thinks might be a waste of time. Anyway, let's just take you through this data. What we do is we have the pulmonary vein, the multi-regional tumour biopsy. We count the number of CTCs in that pulmonary vein. In this, I'm going to show you the data from the next 100 patients and ask whether it predicts recurrence. It shows you again that they're out and about early. And I'll also show you the data from a case study where we looked at these early circulating tumour cells and asked if they are genetically linked to relapse. So this was 100 prospectively recruited patients at sites where the PV-CTC protocol was available. So what we found in 48 of the 100 patients that had at least one of these pulmonary vein CTCs, both squamous and adenocarcinomas. But what we find with a cutoff of 7 is, again, we confirm that those patients who have those early CTCs coming out of that draining vein in the cancerous lung, those are the folks who have the high risk of recurrence. So we still need to do further validation on this to make it useful, but I think it has the potential to inform the frequency of MRD monitoring with CTDNA, come back to that later. But what is fascinating is not all of these CTCs have a tumor matching copy number alteration profile. Many are actually flat. So not all of these cells may be obviously tumor cells. Whatever they are, they predict which patient is going to relapse. So I just take you through the next set of slides. This is a case study. This is a patient who was diagnosed with invasive adenocarcinoma. They had their surgery, they had treatment, it didn't work. They had a metastasis 10 months later after surgery. So we had 28 of those pulmonary vein circulating tumor cells that we looked at one by one. Charlie Swanton and his team had 3 regions of the resected tumor and also the metastasis to sequence. And what is fascinating, so I'll go through this. If you see a brown bar mutation is present, a grey bar mutation is absent, a white bar no reads. And these are the three regions of the resected tumour with clonal mutations, subclonal mutations. And these are three of those pulmonary vein single CTCs. And again, you see some of the clonal mutations, you see the subclonal mutations. But what is interesting is you see a whole bunch of mutations that you only see in the CTCs, so-called private mutations. Now, when you sequence the metastasis arriving 10 months later, they're the clonal mutations, there are the subclonal, and there are those private mutations. And you look at this in the round, 79% of those CTC mutations are in the primary tumor, but 91% are detected in the metastasis, arriving 10 months later. So these early CTCs, it would appear, are the seeds of this metastasis. And you can do a phylogenetic tree, which is what Nikki McGranahan does so well. And it's interesting that mutations shared between these early CTCs and the metastasis included a putative inactivated driving mutation, LZTS1, which is known to inhibit tumor migration. So the lower expression of this tumor suppression is linked to poor overall survival, kind of validating the approach. And when we think about the mutation panel for CTDNA, as well as the clonal mutations, it may be well to put in some of these private mutations, because in this patient at least, that clone that went on to form the metastasis was really difficult to find in the resected tumor. So let's go back to, now let's back up a bit to minimal residual disease monitoring. I just wanted to mention our COMPASS trial, which has been hit really badly by the pandemic, but will be up and running again soon. And this is just the approach for those stage 1 to 3 patients post-surgery with curative intent. The idea here now is to measure those CTCs in the pulmonary vein, and that will tell us how frequently to do our CTDNA analysis. And we're going to go for monthly blood sample collection, not in hospitals, not in doctors' surgeries where the NHS is really busy, but out in community pharmacies. And many of you will know, actually, I was a pharmacist as to start with. So I have enjoyed going out to the community pharmacies and talking to pharmacists and getting them involved in this research. It's been a kind of a whole circle of activity in my life. We'll have blood and tumour samples processed and banked. The tumours will be profiled, the bespoke CTDNA designed in the Swanton lab, including some of those CTC private mutations. And the idea, of course, is to ask whether monthly blood collection is feasible in the community. Will people do it? Will it pick up relapse sooner? We'll do in parallel our immune landscaping of our resected early tumours, looking at radiomics too for prognostic markers. There are parallel trials of earlier intervention with targeted immunotherapies about to start or ongoing. But we're really interested in the feasibility of really collecting blood samples in the community, and this was all completely kiboshed by the pandemic. These are our Cancer Research UK vans who are going to be driving around these pharmacies in North Manchester, picking up our blood samples and bringing them back to the lab. So let's now go back to the topic that I'm supposed to be talking about, which is early detection. I want to take you back to 2017 and the news at the BBC, which with thousands of lives across the UK could be saved after a scheme in which smokers received CT scans in shopping centre car parks. The last summer, this was in the summer of 17, some smokers and ex-smokers, 55 to 74, in these really deprived areas of Manchester, places where you're a little bit worried if you park your car. These are the folks who were targeted for the free lung health check at shopping centre car parks, followed by an on-the-spot low-dose CT scan. And I guess the things that are important here is the word free and the absence of the word cancer. So we didn't know whether this was going to work. Actually, it was hugely successful. Folks came to these vans for their scans in their hundreds and now in their thousands. And in this pilot study, there was a 4-fold increase in diagnosis of potentially curable cancers. So the patients that would have normally been discovered at stage 4, which is, as we all know, incurable, reduced from 50% to just over 10%. This was the pilot study. We're now looking at tens of thousands of folks with this approach. Half of the folks who came in the pilot were offered an immediate CT scan. 42 cancers were discovered, most in stage 1, and 90% of those were offered curative treatment. So this was a turning point that it showed us that this community-based CT scanning was really important, could be very successful, and has become an NHS service in North Manchester this year. So this is what it looks like. The physician invites the individual for a free lung health check. The Low Dose CT scanner vans are out in the car parks of the supermarkets where it's easy to park, in the most socially deprived areas, as I mentioned. And this is what happens. The blue bars are the stage of lung cancer that's normally diagnosed, so stage 4, and the orange bars, and look at the paradigm shift to stage 1 through this process of low-dose CT scans in car parks. But 50% of those scans were false positive, and that patient went through an invasive workup to find out that they'd had a false positive scan. They're expensive, and you can't keep scanning people all the time. So this is where the question becomes, will a liquid biopsy support the molecular definition of cancer? Well, that's clearly the case. Will a liquid biopsy, and I would argue CTCs and CTDNA, identify CT scan true positive patients? Can a liquid biopsy detect cancers earlier than a CT scan, and will we be confident enough with it to do something about that? Liquid biopsy, of course, is readily repeatable to confirm and monitor, and that's where I think the pharmacists and the community come in, because we could have potential for a routine blood-based screening in the community. And I just want to talk about the work we've been doing in my center on ctDNA methylation next. We know that DNA methylation of cytosine at a CPC site is an epigenetic marker. Cancer-specific changes in methylation typically occur early in tumor development. Aberrant methylation occurs at a much higher incidence than somatic driver mutations. And genome-wide cell-free DNA methylation profiling can show an up to 100-fold increase in sensitivity compared to mutation detection. And that was a really lovely paper back in 2018. So how do we do it? There are several methods for high throughput sequencing to look at methylation. The old-fashioned method really is the methylation microarray. So you need fixed probes and you need a large amount of material, not useful here. Commonly bisulfite sequencing is used, which converts all unmethylated cytosine to uracil prior to sequencing. It's technically difficult, it's noisy, it's expensive for the whole genome, and it requires a relatively large amount of material. So I don't think that's the best way to go either. And what we're and many other groups are doing are methylation capture sequencing. So this is a pull down targeting DNA with methylated cytosines, either with a methyl binding domain protein, which is what we're doing, or by immunoprecipitation. And it gives information about methylated regions across the whole genome. But I guess what I'm most excited about, and come to at the end of the talk now, is the work we're doing with Peter Kuhn. And his platform for CTCs is called high definition single cell analysis, and we call it the no cell left behind method. So instead of enriching for CTCs, which always loses cells, and you can't afford to lose them when they're so rare in early disease, all nucleated cells go on a sticky slide. You can then store them in a freezer, and then you can thaw them and bring them out when you're ready. You can develop different multi-fluorescent assays to detect the cell types in there, and there are many and varied. And then there's a big computational challenge which Peter and his team have overcome to pick out those very rare cells, which can then be picked and sequenced. One of the last things I just wanted to mention is nanoscavengers for biomarker discovery. And this is work with Lois Gardner, Kostas Kostorelis, and Marina, I can never pronounce her name. It's a very complicated Greek name. Certainly at the end of the day, it's impossible. But what they use is nanoparticles, and they look at the corona. So these are nanoparticles that scavenge blood, and there's a surface capture of low molecular weight, low abundant proteins. So this problem of low abundance proteins being swamped by high non-specific, high abundant molecules, that is the poor signal to noise bête noire of proteomics, is now overcome, because you can now go with the corona. This is the signal to noise you see normally, but with that corona analysis of the nanoparticles, you can really begin to see low abundance proteins. So I think this has got huge potential for early detection. And we asked with our colleagues at the nanomedicine labs, at the graphene labs in Manchester, can we do the same for ctDNA? And again, there's not a final story here, but this is our first publication in Nanoscale Horizons. And we know for sure that there is circulating free DNA on this nanoparticle corona. And of course, the next question is, will that give us sensitivity for circulating tumor DNA? And can we combine that with Max Dean's incredible assay? So what does the future look like? This is the last slide. You know, we've talked about the cancer tests in supermarkets. We've talked about liquid biopsies, early detection of minimal residual disease. The liquid biopsies will support the low-dose CT scans. We'll hopefully reduce false positives. We'll use them for risk of recurrence. And I think community is where we need to go. And I think this is what it'll look like in the future. It'll be a reminder on your smartphone. It's the first Tuesday in the month. You need to go to town. You need to go to your community pharmacy today for your cancer screen to give in your blood sample. Just wanted to mention, I was privileged to be part of the group that wrote the Early Detection and Diagnosis of Cancer Roadmap to the Future. It's worth a read. It's now a Cancer Research UK priority, as many of you know. And a final observation. So should we fish for liquid biopsies for early detection in a pond, high-risk cohorts? I would argue that's a good place to start. Or in an ocean, pan-cancer, that's where Grail's going. They've got more money than me. Encouraging progress to date with a refocus of funding, I think. A real fishing trip or hypothesis-driven, scientifically rational biomarkers, that's the challenge. And to get to that, we need early disease biology, and we need better models to get there. But what I think is really important is how much of a team you need to reach the summit of early detection. There's too many people to mention. They're all there. My team is wonderful, as is the TracerX team. So I'll stop there, and hopefully we've got a little bit of time at least to have some questions, although I do have to run off by about 6:05 at the very latest.
Thank you so much, Caroline, for this tour de force talk. Fascinating. I really learned a lot. So we have exactly 15 minutes for questions. And
crack on.
Really looking forward for questions from Sylvia. It correlates a lot with your talk, and from John and from Andy. Yeah, go ahead, anyone. Who wants to start first?
I start. Privilege of females. Caroline, this is fantastic as usual. It seems to me that you're putting all together the research question, which is your last comment. You know, we need to understand the biology of the disease with the more healthcare type of thing. Yeah. You know, my interest is in the first, and I would like to, and although I totally agree with the fact that we need to go to the community and use the community for what it is. I've tried desperately. In Italy, we have a truck for frozen food that goes around. You know, it's a very common supermarket thing. They go around all over Italy. And I've tried to talk with the CEO a couple of times, trying to make him use the trucks to ship blood. And so far, I haven't been positive in the backup, in the response, because he is skittish about cancer. But I think that things are changing. So you give me a good idea.
Nobody can, that's for sure.
Yeah, I know. Thank you. So I'll try again. But back to my question. So I'm very much interested, because I have become in the early part of your, you know, is the early disease the same? Why are there, and it's very clear in colon cancer, it seems that in lung you are seeing the same, that it's not linear, that it's not true that there is, you know, this linear escalation in the evolution. There are tumors that, as I call them, they're born to be bad or born to be good. And this is a big liquid biopsy or CTC, whatever. It's really a very good opportunity to figure out who they are, so that we can study them. So I wonder if you feel that this is it, and if so, what are you doing?
Yeah, that was entirely the motivation for the single CTC sequencing. You know, we have to do so much more of it. It's really expensive, so I'm waiting on a big grant. But I think we need to have a really good understanding of the heterogeneity of those early CTCs. So are there some which are born to be bad, as you said? Most of them are going to die in the circulation. Let's face it, that's what happens. So, you know, can we get any sense, if we do enough of this science, of the ones, you know, not only do you see those early CTCs, you know which ones are going to cause trouble. And I think that's a really interesting question, and one we need to just explore more.
Okay, let me ask you a second question about this. Why there are tumors like lung or breast that are producing CTC, and other tumors that don't, like colon, colon? Is it
Yeah.
just an anatomical thing, or is it a biology?
So
Because
yeah,
this-
that's when I talk about this all the time. So we tried really hard, as you know, to look in colorectal cancer using cell search, you know, and it's just the worst technology for colorectal. You can't find them.
yeah.
But something must be there, right? Unless all of that's going through lymph, there must be cells that appear in the blood that go elsewhere, and they go into the liver, right, in colon, they grow tumors. So, you know, I think about my father, who's had a tumor the size of a grapefruit removed from his colon 12 years ago, right? I mean, there was no evidence of any metastasis at the time. He now has evidence, unfortunately, of liver metastasis 12 years later. So what were those cells up to? You know,
Yeah, well, this is another phenomenon. I think that we're going back to the second question. I have another question, but see if there is somebody else that asking. John.
Oops, you're on mute, John.
You're mute, John.
Well, you were asking about, you know, what's been happening to those tumours while they're sitting and you can't find them. So it's a fantastic paper which has come out in colon cancer. They enter into this
diapause, yeah.
They go into this sort of embryonic state where they sit and go to sleep for a very long time, and you won't find them.
Well, that's what the dormancy is again, isn't it? So you know, the grand challenge, one of the CRUK grand challenges is dormancy. It'll be really interesting to see what they come up with.
Yeah.
Yeah, but the point. Sorry.
That's where I agree with you. Looking at the biology is really important. But I wanted to ask you about follow-up, really, and the role of sort of what would you call it? Just, you know, using, as you said, using the telephone, using a contact between the patient so you don't lose them afterwards.
Yeah, and that's really challenging, you know. So we found that in TracerX, you know, you get them, they have their surgery. Getting a relapse biopsy is really challenging.
Right.
Just keeping track of where they all went. Now in the NHS, at least that's doable because we're moving towards electronic health records, you know, and the digitization of healthcare. So it will get easier, I think, but it is definitely a challenge. And that's why I wanted to do the feasibility study. So would someone go shopping and once a month pop into their pharmacy and give me a blood sample? That's the question. And I would know the answer if there hadn't been a pandemic, which really messed it up.
So that's my other question, which is kind of psychological and sociological. I mean, do you tell them that this is a test for cancer?
Yes, I think eventually, I think you do. I mean, we aren't able to report anything we find in the pilot study because we don't have a validated test yet. But I think you do, and I think if that's where we've involved psychologists in the study, because you know everyone wants to they want to have their surgery and not think about cancer ever again. But once you start explaining, well, actually, what this test will do is reassure you that it's not coming back. Because, you know, for most who've had early surgery, that is the case. So I think it depends is how you do it. Challenging. All of this is challenging.
I'm glad you brought the dog in.
Which dog?
Sniffer dog.
The sniffers.
Caroline, really nice talk. A couple of things. One is I was just thinking whether how quickly the cells regenerate in certain organs. So the digestive tract perhaps has a faster turnover, and whether that, as well as dormancy, might also partly be an explanation for why we're seeing differences in lung and, say, colon. But the other thing I think is lovely is the fact that, as other listeners have commented, you've got the quite nice bench to bedside, you know, sort of community scanning approach to this, as well as all of the upstream careful biology approach. Do you think that this could actually be a bit of a trailblazer for how we might actually get some really decent real world data using a community approach, which should actually be considered more by regulatory bodies and HTAs to sort of lift us up, so that we don't just rely on randomized trial data, but also have this as very much an important adjunct? Just interested in your views.
I absolutely 100% agree with you, Lisa. And, you know, one of the big areas in our Manchester Cancer Research Centre renewal grant, everybody's writing grants, big grants at the moment, is real world evidence and how we use it to do exactly what you've just said. So yeah, I completely agree with you.
Yeah.
I mean, the really frustrating thing as a translational scientist in clinical trials is you have to wait forever for the trial to finish. Then you have to wait forever for the statistician to clean the data. And even then you get sent a file that it looks like it might as well be in Greek because they've coded it in such a ridiculous way. So, you know, if you're the translational biologist trying to understand the biomarker and the biology and what that biomarker might do for the patient, you know, you have to sort of wait for about 12 years before you get the data. Drives me around the bend. So the more real-time, real-world, iterative approach, the sooner we can get to that, the better, in my view.
Accessible, isn't it? And also it makes the patient more at the center of all of this, in a sense, because it kind of gives them a bit more of the onus and the opportunity to feel as though they're doing something proactive, I guess.
Yeah.
So,
and the real challenge here is money. So these early detection studies cost so much money.
Yeah.
So we could get the pharma, I'll say it before John does, we could get pharma interested, you know, and all of the money they have, if they could get much more interested in understanding early biology, because that's where their targeted agents and their immunotherapies might actually work better. If we can build all of that into the messaging, I think that's the way to, you know, we've got to get big funding to do this properly.
Well, I just read today, I shared it with Lisa, you know, the KRAS inhibitor from Amgen.
Yeah, I saw your LinkedIn post, John.
Right, $18,000 a month. You know, you look at, and so these people have a PFS of about 7 months. So just work out the sums and imagine how much money you could actually put into a program of early detection. I mean, you know, this is where the agencies.
This is the problem, you know. So, you know, the Cancer Research UK have done a really good job of bringing early detection priority right up their list.
I agree.
But you know, the biggest grant you can get is still about 800,000, and it just scratches the surface. You know, I mean, I'm very grateful for it, of course, but I need millions, not hundreds of thousands.
Yeah, I agree with that, but you know, I also see trouble brewing in the horizon of liquid biopsy, because, for instance, the two main, I don't know in Ivata, I don't know what Nizan is doing, but in Ivata, Garden and Natera are selling 1 point of a liquid CTDNA assay at you know, anything between $4,000 and $5,000. So I mean, and we, for sure, I can tell you that you need to do at least 3 point in the MRD in colon, which is a big shedder, just because to outdo the negative, the false negative. So that's 10,000 just to begin with, and then you have to do the follow-up. And if you want to do a monthly follow-up, this is going to cost an arm and a leg. Now, what is the real cost of all this? Certainly not 3,000.
I can do it much cheaper than that, Sylvia, for sure.
I know, but that's where, and I can tell you that the industry. I'm designing I have designed the new Pegasus, and I'm looking and asking to pharma to give me the drug for free, because I'm bringing targeted therapy frontline in adjuvant in colon. They're very, very interested in doing that, because they have finally saw that what they can do is use, and all of them, Roche to begin with, is getting into the market of liquid biopsy, as you know, with the Foundation Medicine. So this is going to be where they're going to make a lot of money out of it, and they think that they're going to do that. So we have to be prepared, because it's not going to solve the problem of the cost of the treatment, not even in the early detection, and certainly not in the MRD and the monitoring.
I mean, I agree with you. I think, you know, the biggest win will be for the patient who can have surgery with cure. I mean, this is the most important, right?
Yeah,
absolutely.
Absolutely.
It's a longer, it's a lower.
You know, the challenge is then becomes a different one, right? So now we don't have enough radiologists, we don't have enough surgeons, you know, so there's trouble ahead, whichever way you look at it. But I think ultimately, you know, this is where it's going to end up.
Yeah.
Well,
I mean, I totally agree with that. I think this is where, you know, surgery and radiology are going to be really important And I just don't see how pharma are getting involved. I mean, the scary thing from your group, Sylvia, is from the Bardelli group, was if you give an EGFR inhibitor to tumours, it increases DNA instability. So, you know, even if you get in very early to the first mutation and you think, you know what the founding mutation is, and you give some sort of targeted therapy, that cell will then adjust, and the therapy could be worse. And this is where I think surgery and better imaging and finding micrometastasis.
And possibly
immunotherapy,
you're only how to use it properly.
Now, John, you're not going to treat anybody that these are already metastatic patients. You're not going to touch the ones that are not. You're not giving it preventively in a neoadjuvant fashion. You're only treating patients that have residual disease that have something. So those patients are doomed to fail in any case. So I don't know. We're doing the randomized trial because we want to know what is best to do.
But if you detect a stage 1 disease and you have surgery, right, and then you do minimal residual disease monitoring, the second you see a signal and you have some idea of what immunotherapy might, you know, in a low burden setting, is immunotherapy going to work better? I think that's a testable hypothesis, and I think it's a good one. In
colon, I don't know, because
I don't know either.
They're cold tumors, so you know they're immunotherapy.
Well, you're right there. But,
I'm also looking at, I think we should also look at another, what we're talking here, it also has to do with your father. You know, there are some permissivity that it's of all kinds. The immune system must be permissive. The vascular system has to be permissive because they have to go out of the tumor mass. So for instance,
that's another really interesting story. So the most exciting biology we're doing at the moment is vascular mimicry. How tumors make their own blood vessels is absolutely fascinating.
Absolutely. And there are some very interesting data. Perhaps next year I can talk about this. Because I think that's an important moment to catch the horses before the corral. It's you know, before you close the corral. So you have to really catch them at the moment that they're getting out, because once they are out, as you suspect,
before the horse has bolted.
But before the horse is bolted. Sorry, my English is really very.
It's wonderful. And it sounds much nicer than my English too. I much prefer.
I was pleased that Caroline used some French.
When?
Bête noire.
Oh gosh, yes. Well,
bravo.
Indeed.
Okay.
Indeed. Do you know, guys, I'm going to have to go in 2 minutes.
Yes. On that note, I'd like to thank Caroline and thank you so much for your passion and amazing discussion with Sylvia and John. Really, really great talk, and I think we cover the whole perspective from basic research and all the challenges that you just explained, and how you don't see this getting to patients, and my side of the story where we are trying to get drugs to patients through HTA. And it's such a complicated and a long journey, and everything you've said, there's amazing progress, amazing possibilities, but we really need
Well, unfortunately, I'm not sure I'm going to get this experiment done before I retire, but anyway, we'll see.
Well, we need to fix the system to make things happen faster. So everyone, have a fantastic evening. Caroline, thank you one more time. Bye-bye.
Thank you.
Bye, everybody.
Thank you, guys.
Bye. Bye-bye.
Ciao.